Hi, also last video for today. We're going to be going over the scientific method and a little bit about the procedure that we would do analyzing information in a lab. I'll explain it briefly. I won't go into super big detail because it is related to formula and math. Because we'll be doing it in more detail when we actually get to our first lab, whether it be virtual or in person. So let me screen share. It would be helpful if you guys had your journal or at least a piece of paper that you could take notes on. Um, you can choose to pause before or after I finish talking about this slide to kind of jot down this diagram into your notes because it's a pretty useful one and it has a kind of brief explanation underneath each section or each portion of the scientific process. So I'm gonna go ahead and explain it. You can pause it at any point to kind of copy this down. So the scientific process generally begins with observation, right? So if you notice something that's kind of weird, you notice something that you don't know the explanation to, you're going to want to find an explanation for it, right? That's naturally human inquisitivity. You don't know something, so you need to find out why it's doing that or why it's like that or how it's doing that, right? And human curiosity is the whole reason why we understand a lot of things because people made observations. They were just like, what the heck is that? And so they went along to kind of test out how things became the way they are or how things were made or how things work the way that they work. Like, for example, like gravity, somebody one day was just like, why do things fall? And then they went about testing it and they were able to deduce that it came from things being attracted when they're of different masses, right? So a hypothesis is after you've observed this phenomenon that you can't explain. You look up sources to see if anything or anybody has ever made research or conducted research on the same topic that you're kind of interested in looking at. And you formulate a testable statement. So a lot of hypotheses come from if-then or are structured as if-then statements, right? For example, back to gravity. If I hold the apple up high and let it go, then it will fall to the ground because of gravity, right? That's the easiest way to formulate it. We're actually going to talk about a different kind of hypothesis called a null hypothesis later, but a hypothesis is usually the statement that you're going to be testing, right? You're like, if I do this, then this should happen. Okay. After you formulate your hypothesis, then you can actually go ahead and conduct your experiment. You can't just conduct an experiment without some kind of guiding statement, right? That's the whole purpose of the hypothesis. So you do your hypothesis and then you conduct an experiment that is going to either support or fail to support your hypothesis, right? So you either, it can be an observational experiment, it can be something that you're actually manipulating, right? Depends on what kind of scenario you're using, right? If it's an environment, usually you don't manipulate that because that affects a lot of organisms and their lives. If it's on humans, you usually can't test that many things on them. There's like a huge number of FDA guidelines on that. But if it's something that's like not related to living organisms, or if it's something that doesn't really harm anybody or any organism to do, then usually you can manipulate conditions, okay? Based on the results of your experiment, you make a conclusion. Either your hypothesis was supported by the experiment or it wasn't supported, right? So maybe it's because your phrasing isn't right. So then you go back to the drawing board and you come up with a new hypothesis on the same topic. And you're like, maybe if I phrase it this way, then when I conduct the experiment again, it might support it, right? That being said, this little loop right here that you see on hypothesis, experiment, and conclusion, it's not always just a direct loop, right? Sometimes you have a hypothesis and you do the experiment and you do multiple rounds of the experiments to make sure that maybe if you only did one, the results are just random chance and they're extreme to one end or the other, right? If you do a lot of experiments and you kind of average out the results, then you get the like average of maybe normally if I were to do this and there wasn't any extreme case, then it would or would not, then the experiment's results would or would not support my hypothesis, right? You don't want to just do it once or twice because again, the chances of it hitting like an extreme end of the spectrum of supporting or not supporting, it's like 100% or 50% at that point. You want to make sure that that's not the reason, right? You don't want an outlier like you talked about in math. You don't want an outlier to be the reason why you're like, oh my God, my hypothesis totally worked. right? Because then you could look into literary research and be like, wait, but none of these people's experiments that are pretty much the same as mine have the same results or have the same explanation or they have the same conclusion, right? That kind of gives you an indicator that maybe something went wrong in your experiment. And you can also explain that in your conclusion. When a hypothesis is tested multiple times, like thousands of millions of times with experiments, and the conclusions all come out to be the same, then you get a scientific theory, right? That means that basically we know this to be true in our plane of existence, right? Nothing's proved it wrong, but there's no way to 100% prove it correct either, right? Some things are scientific theories and some things are scientific laws, right? For example, theory of evolution. We have yet to figure out if there was another possible way that we came to be how we are now, but there hasn't been an alternate explanation that's been supported by the evidence that scientists and geologists have found throughout time. So it's a very, very strong theory, right? Nothing really opposes it, but there's no way to 100% concrete prove that it's true. Okay, so if you've copied this down, we're going to move on. If not, please pause and then copy this kind of little diagram down. So a scientific theory, not a hypothesis, is usually broad in scope. So it's very, very general. That way it makes it more applicable to a whole subject, for example, and then it's also less likely to be wrong. So a statement that is more broad and applied to many things and is right over many things is a scientific theory. So the example would be for broad in scope is like saying science, when in reality there's biology, there's chemistry, there's physics, there's anatomy, right? There's a lot of kinds of sciences, but broad is just saying science, okay? The scientific theory is supported by many different observations over many different years, right? So just because time changes doesn't mean that the theory becomes wrong, right? The theory must maintain over many, many, many years and over many, many years of experimentation, okay? So it's the closest thing to a fact that we have in science. Like I said, it's not 100% true because there's no way to prove anything 100%, right? Because there's always like outlier results in experiments, right? But it might be 99.9999999% true. And that's the closest we can get, right? Because there's no significant evidence against it, right? Example is like cell theory. All organisms are composed of cells and cells only come from pre-existing cells, right? You're made of cells. You're a living organism. And you came from cells from your mom and your dad, right? The egg and the sperm, those are cells, right? Bacteria also come from other cells, right? A bacteria will split off and become two bacteria, right? There was originally one bacterium though, right? So the cells are coming from another pre-existing cell. Homeostasis is another theory. The internal environment of an organism stays relatively constant within a range that is protective of life, right? If, for example, your body temperature gets too low, you're going to get hypothermia and you're going to die. If your body temperature gets too hot during a fever, your brain can start malfunctioning because of it and you're going to die, right? For example, bacteria, when they're in like a saltwater solution, they got to be able to maintain and pump water in and out as they need. Otherwise, if they get too salty inside or don't have enough salt, then they're going to die from it. Gene theory is organisms contain coded info that dictates their form, function, and behavior, right? We all know what DNA is at this point, right? Those are our genes. Bacteria have DNA, or they have genes. Plants have DNA, they have genes, right? Some organisms don't have DNA, but they have RNA, but those are also their form of genetics, right? Without this kind of code of life, the thing is not alive, right? For example, rocks don't have DNA. Rocks don't have RNA either. They're not alive. They're abiotic. They were never alive. Okay. That being said, if something is dead, it's not alive, but it was alive and it did have DNA. And you can even find DNA traces inside of the organism cells even after it's dead. Okay. Number 4, evolution. Like I said, all living things have a common ancestor, but each is adapted to a particular way of life. So over time, things change because the environment changes, right? And the best suited organism to that environment is the one that survives. So then maybe the organism that exists 30,000 years down the line in the same area lives in a swamp, but the one in its ancestor that lives in that same area lived in a desert, right? So they're in super, super different conditions. That's why they look different, but they have genetics that were passed down all along. And each time there was a new mating season or a new offspring, the ones that best fit the current environment from desert to swamp transition was the one that survived the best and produced the most offspring. Okay, a hypothesis is narrow in scope, right? We just said scientific theories are broad. Hypotheses are narrow. It's really hard to test something if it's really, 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 really broad, because then you have a lot to test, right? You want to make it super, super narrow so that you can test it easily inside of an environment like a laboratory setting. Okay, you have to be able to test it. If you can't test it, there's no point in even making a hypothesis, because then you can't do an experiment on it. There's no, if you can't work with it, why'd you make it, right? It must be able to be proven wrong. Okay, so if you can't say that it's supported or not supported by your data, then that's not a hypothesis. You have to be able to say the experiment's data supports it, and in conclusion, the hypothesis, maybe you need to go edit it, or you may need to go back to the drawing board, right? Or you can say it is supported, and then we're going to go back and do more experiments to see if it is supported, right? You have to be able to show that it is correct or not correct. If it's a statement that is already proven, then it's not a hypothesis anymore, right? Then it becomes scientific theory. Okay, so what I talked about earlier is the null hypothesis. So I do want you guys to write down the null hypothesis in your notes if you have time. So I'm going to do a quick explanation overall, and then I'll give you time afterwards to write down your own version of an explanation. So how we write it is we write H, and then in a subtext, we write a little 0. That's not an O, it's a 0, okay? So that 0 is pronounced not, like without, okay, as a synonym. So H naught is representative of the null hypothesis. So we'll write like H naught and then colon, and then we'll write out our statement of the null hypothesis. And basically it states that what you're testing will have the same result as a control, right? Whenever you make an experiment, you have a manipulated set and you have a control set, so that you have them two to compare with, see if your manipulated set that you changed actually made an effect and made it different from the control, right? But the null hypothesis is always going to be, there's no difference, right? Even if you change this, there's no difference. The null hypothesis is always that. Your hypothesis statement, if-then statement, might be different from the null hypothesis, right? You might be saying, if I do this, then this will change. But the null hypothesis is always going to say, there shall be no difference between these two groups. Okay, so if I have a battery that's not turning on, if I have a flashlight that's not turning on, and I take it out, and I'm like, maybe I need to change the batteries, right? My null hypothesis and my hypotheses are going to be different. My hypothesis is, if I change the batteries, then the flashlight will turn on, right? But the null hypothesis, as listed in this right now, is saying the results of changing out the batteries in a non-functional flashlight were no different from the results of not changing out the batteries, right? So the null is saying, I change out these batteries, they still don't turn on, right? But my original hypothesis, the whole reason I was changing the batteries in the first place was, I think that if I change the batteries out, it's going to turn on, right? There's a lot of other scenarios that you can work with a flashlight. It's not always, sometimes it's the bulb, sometimes it's the batteries being flipped around because they already work originally, or maybe it's because one of the ends of the electrical connection of metal to metal is blocked, right? But a null hypothesis is always saying there's going to be no difference between what you change and what was originally the case, or what the control is, okay? So this is kind of off of Google. A null hypothesis is a hypothesis that says there is no statistical significance between the two variables. It is usually the hypothesis a researcher or experimenter will try to disprove or discredit. So if you're able to disprove a null hypothesis, then it means that there is a difference. No matter how big or small it is, there is a difference, and that's important, right? Because that means something changed, something that you manipulated caused an effect. And that's honestly what all science is looking for. So chi-squared test is an easy way that a lot of scientists, especially biologists, use. It's basically a statistics test, actually. So if you ever take AP Stats with Dr. Bird and you see this, you're gonna know what it is. Or if you already took it with him, then you already know what this is, right? So chi-squared is the sum of the squares of the differences between what you expected and what you observed. And that might not mean anything right now, but it'll mean something when we actually put it into play in a lab. So for example, if you were expecting how do we say this? So if you were expecting, this is usually numerical data, obviously, because it's a formula. So for example, if bugs are crawling around and you put food on one side and you expect the bugs to go over to the food more, and you say, we expected more than 50% of the bugs to be on the food side by the time we counted them. But when you observed them, it was 50-50, right? Then your difference is going to get squared and then divided by your expected, which was the over 50%. And then you add all those up for each different group of experimental groups that you have, right? So the control is always going to be 0, but you might have multiple experimental groups based on different food types that you're testing, right? For example, so maybe one of them seems that like for fish food, if you have bugs crawling over, you might have more than 50%, but when you square them after you subtract them and then divide them and add them all up together, the number might not be that big, or it could be really big, right? Each round that you do is going to be different. But when you average them all together and you use the chi-square table, you're going to look at degrees of freedom. So degrees of freedom is related to how many groups you had. So if you tried 4 different foods, you're always going to go 4 minus 1 is going to be the degrees of freedom. So you look at 3. And we always use 0.05 in biology for the p-value. It's just for 95% error, just in case, right? So if you have 3 and you have 0.5, you go draw along the lines. And 7.82 is the standard that we're going to compare to. So if our chi-square that we calculate using this formula is bigger than 7.82, that means we had a difference, right? That means we can reject the null. That means that something that we did in this experiment has caused the bugs to not act like just randomly moving around, just like a control, right? But if it's lower than that, then we fail to reject our null hypothesis. It means that even though there was a difference, it's too small. It doesn't really matter. Okay, so you can do labs, the same lab, many different times, and you can get different results each time. Sometimes they can be significant and cause a difference, and sometimes they won't. Right, so that's one easy way to test for a null hypothesis. Again, it might be confusing now, but we'll actually explain it when we get to do it in our first lab. Okay, so an assignment that I have for you guys to follow up on this, it's already in your Google Classroom. You're going to read page 13 in the AP Bio textbook. If you've already logged into the McGraw-Hill Connect and gotten into the class, then you have access to the entire textbook. I've also attached it in Google Classroom to make sure that you can read it, even if you haven't been able to access it yet, or if you're having issues, email me if you are. And then you're going to write a short essay response about the purpose of statistics in scientific research, right? Why is it important? Why do we do it, right? Why are you putting math and science? How are they related, right? All of that short essay response, and then submit it in Google Classroom. I'll have it graded, and then I'll be up in focus. That's all I have for this. If it was confusing for you, you can rewatch this multiple times. If you are still confused on the chi-squared part, I would hold off on worrying about it. Just make sure you get the notes on the scientific method down. Okay, that's it.